Thank you for coming to the Biogeochemistry Science Friday at this early hour. We have two guest speakers from Asia today, and so we are accommodating some changes in the time zone. First off, we have Jianyang from East China Normal University talking to us about plant functional traits and carbon processes. So go ahead, Jianyang.
Okay, thank you. First, I want to thank Forrest for the invitation, and also I'm Jianyang from ECNU, and I'd like to thank all the co-authors and also the modelers from the MIST-MIP and the CMIP project. So today I want to talk about how we can use the plant functional traits and the carbon processes to evaluate the model based on traceability framework. Next, okay. So we know the land carbon sink now is offsetting about 30% of the anthropogenic CO2 emission, but its dynamics in the Earth's model are quite uncertain. So these figures shows that the IPCC report have a large uncertainty on the land carbon uptake from the AR4 to the latest one, AR6. Although the AR6 didn't report the carbon uptake rate, but we use the land carbon storage. It's similar. So we can see the model and the model is disagree with each other in the historical dynamic. So it's very happy to see that the ILAM results in the IPCC AR6. So I select the land carbon part. So the color in the purple shows a better value, and the orange is a worse value. So you can see that the model performance has been improved from the CMIP 5 to CMIP 6. When we look at the net ecosystem exchange or the net exchange of the carbon between the land and the atmosphere, we can see the model data difference didn't improve a lot. It's a small improvement. And when we look at the model-to-model difference, it's still very large for most of the carbon variables. So we think how to further improve this model is still challenging. So recently, a few very good reviews have pointed out these issues, like the ICHIS and the TED review on GCB. They point out that the improve the parameterization of the carbon processes could be a very important future direction. And also, like the right figure is from the recent review paper from Anthony Walker. He used in this figure, we can see that the plant function traits and affect many carbon processes in when we start simulate its carbon cycle to the elevated CO2. So we know the plant function traits are fundamental parameters in these models. So how we can reduce them is how we can improve the parameters of the traits in this model is still very challenging for us. So one of my earlier studies shows that the current models usually use the plant function traits to define the PFTs in the models. So this is two important traits. One is a specific leaf area, the right one, the Vc max. You can see this in these models, they usually, or we have one model usually have very similar or identical trait for one PFT. Also in the same region, different models usually have very different trait parameters. So the key question is, can we reduce the model uncertainty on land carbon cycle by improve the parameterization of plant functional traits? This question we can ask separate into two specific ones. First one is, can we link the plant traits and the land carbon cycle for model evaluation? Second one is how we can improve the model parameters based on data. For the first question, we recently developed a tool called Traceability Analysis. In this framework, we developed from a matrix equation. The basic idea is we first figure out there are several traceable components in the carbon cycle, which are shared by different models. Then we compare these components to understand why these models are different. So as this figure shows, the plant traits not only can affect the carbon uptake, but also affects the baseline carbon residence time. So we first apply this traceability framework to the CMIP6 models. So as shown in this figure, we can, in the historical runs of CMIP6 models, on the global scale, you can see in most regions, the model-to-model difference is results from the baseline carbon residence time and the GPP. And then when we look at the mean value of the global or the total value of the global, you can see that the baseline carbon residence time is a major uncertainty source in the current CMIP6 models. So this is historical runs. When we look at the initial state of these models, so the pattern didn't change much. So this is very interesting like that. Before the model do the forward simulation, the uncertainties is already there. So the most uncertainty is result from the baseline carbon residence time. That means that those parameters affecting the baseline carbon residence time played a very important role in the current what we have seen in the IPCC report. So in this framework, when we look at the equation, we can see that like the baseline residence time at the initial state of the 1850 is from three components. One is the allocation of NPP allocation. The second one is the carbon transfer between pools. The third one is the age of carbon in different tissues or carbon pools. So that means plant functional traits in especially the allocation trait and the transfer traits or some carbon age lifespan traits is very important to determine the baseline carbon residence time. So these parameters are very important to determine the difference between models on in the baseline carbon residence time. The second part in the framework, we can see the plant function types, the parameters usually can affect through different pathways to affect the GPP. So there is it's clear in the nature ecosystem like that. In the vegetation, the carbon processes have a propagation loop. For example, the GPP uncertainty can transfer to NPP and then to biomass and leaf carbon and leaf area and back to GPP. So in this loop, the plant functional traits played a very important role. For example, the carbon use efficiency can affect the NPP uncertainty from the GPP, and also like the specific leaf area together with leaf carbon or leaf mass to determine the leaf area uncertainty. So it's very difficult to test apart or trace which trait has contributed to the GPP uncertainty. So my question is, how do the plant function traits contribute to the model uncertainty on carbon uptake in current models? In fact, the GPP itself also has been improved in the CMIP6 models, but it's still there is a still large uncertainty, not only in the CMIP models, but also in the data products used to benchmark or evaluate the models. So I think it's still important to understand why GPP is so different between these models. To do this, because the CMIP model, CMIP6 model, we don't have more information about the trait information or other like specific leaf area, lots of other processes. So we use the MISTMIP project. We focus on the East Asian monsoon region because this is large carbon sink at the middle to low latitudes, GPP uncertainty is still very large in the models as shown in the right figure. So to do that, we based on the MISMIP model, the experimental design. In this, they have the different simulation experiments to do the factorial analysis based on the constant climate and then use of CO2 nitrogen forcing, and then he turn on one by one. We can use this output to evaluate the impact of the three major uncertainty sources. That are the external forcing, model structure, and also parameters. So the results show that in this region, also the GPP has very large uncertainty between the models. And not only, there's also a large special variation of the uncertainty magnitude. So the right figure shows that the lines show the difference between the mean between the constant simulation and the shaded area is the impact of the environmental change. So this figure should tell us the model's difference in this region is mainly from the constant simulation, but not from the environmental impacts. That means the model structure and the parameterization of the different plant traits are very important uncertainty source in these models and also in this region. To do that, we use this loop structure to evaluate different traits. So we collect the parameters and calculate them from each model. And so this is a clear show that the model, the uncertainty is propagating between these carbon fluxes, and different traits play different roles in this propagation. You know, in this region, in the East Asian monsoon region, you can see the specific leaf area is most important uncertain trait in determining the GPP variation between models. So, and then there are some other traits like the fraction of leaf that is determined by the biomass allocation trait between the models. I think this is this result tell us that it's very important to use trait data to constrain plant traits and their parameters in these models. So my second question is how to improve the model parameterization of plant traits based on data. I think this is a very difficult question. So there are many activities to incorporate the trait acclimation and covariance to improve the modeling of carbon processes. I put here an example from a Nature Commission paper, and assuming this paper, assuming this figure, you can see we can incorporate the trait covariance into the model, and based on a specific leaf area, then we can calculate the area-based leaf nitrogen content, leaf lifespan, and VC max. Also, we based the wood density to calculate the mortality rate, something like that, based on the trait covariance. Then we can generate a parameterization of different traits and to do simulation to generate a spatially varied trait combination. So this is a basic idea. I think many models use the traits to simulate the spatial difference of these parameters. So we can see that in those trait-based models, there's two types of plant traits which are very important for carbon cycle. One is related to photosynthesis. This usually a VC max or leaf area leaf nitrogen or leaf lifespan. And also the other type is related to mortality. Usually use the tree mortality rate or stem size or tree size or wood density. So when we look at the leaf photosynthetic traits, we can see usually use the leaf spectrum, leaf economic spectrum to describe the spatial covariance of these traits. So we ask the one question is whether this leaf traits covariance inferred from the leaf economic spectrum will be changed or altered by the environmental change, especially the global change factors. If it not change, I think that we think the covariance is very constant, and if they change, they have two ways to change. One is to change the intercept of the covariance. The second is the slope. So when we usually read their original papers, they usually say there's no impact of climate, environmental factors based on the spatial covariance. So here we did a meta-analysis based on the experimental data. So we collected more than 200 pound species data from more than 500 experiments. So the treatment includes the warming, drought, CO2, and nitrogen. You can see that about half of the data is from the field and half from the greenhouse. So the meta-analysis results show that plant trees are very sensitive to global changes. So you can see the distributions of the trait response is most of them are normal distribution, and so that means we, as highlighted by many previous studies, the trait acclimation could be represented as probability distributions, especially how the joint distribution of these trait acclimations. We also see that the acclimation directions are so different between these traits. So when we look at the species level trait acclimation, so in this figure, one arrow represents one species, all from different experiments. So you can see when the environmental change, so this is one line represents different factors. So the different species can have so different directions of acclimation. So that means once the environmental change, so the species may not respond in the same direction, and most of them are not follow the leaf economic spectrum like the line here. So when we look at the twin species or like over species over space, it's clear to show that the relationship or the covariance between the traits are not changed. Most of there are a few changes that happens on the intercept. The slopes never change. So that means the covariance of between the traits over the space could be robust for derive some correlations between traits to the models. So due to that, based on that, we did a modeling experiment based on the cable model. This is one of the process-based model. It's very widely used now. So to incorporate this data-driven trait acclimation and covariance, so we designed 4 simulations. The first one is the control one. We just use the ambient CO2, and with the derived trait related covariance between specific leaf area and the leaf nitrogen content based on the control plots. The second one is we double the CO2 concentration, but not change the trait acclimation and covariance. The third one is we only incorporate the trait acclimation. The last one is also not only change the acclimation of trait, but also the covariance between these two traits. So we did 4 simulation and use each one to minus the control one. Also similar for the factorial experimental simulation. So the results is first, we didn't carefully study the reasons, but the results show that when we didn't change the trait acclimation and covariance, so we can see a very large CO2 positive effect of CO2 on the MPP. Then when we added the trait acclimation, you can see there are many regions have the negative response. I think that's the reason, because the specific leaf area is reduced under elevated CO2. So the negative results vary from the leaf area, the reduced leaf area in this region. So when we added the covariance, so the positive response come back in most regions, but still in some dry region, it's a very weak negative response. So this case, I think this modeling case show us like that. Although the trait variance is only change a little bit, but the impact on the carbon processes, for example, MPP, and its response to CO2 or elevation or other environmental change could be very large. So this is, I think, a modeling case showing that incorporating trait acclimation and covariance could greatly change the model response of the carbon processes to environmental change. So the second type of the traits, we focus on the mortality traits, because we haven't done modeling studies based on the trait mortality traits, but we have a field site. So we work with data and to study the covariance between the stem size and the tree mortality rate in a 20-hectare plot in a subtropical monsoon forest. So in this forest, in this 20-hectare plot, we have 117 species, tree species, and we totally have more than 150,000 individuals. I mean, during the 2010 to 2015, during 5 years, we monitored more than like 14,000 trees which were died over this period. So then we studied the left panel shows the distribution of all the dead trees in this plot, and the middle panel shows on the species level. You can see those more than 100 species show so different patterns of the trees mortality rate and the tree size. But when we lump them into the community level, it shows a pattern of the U type. So we're using the PCA or different attribute statistical analysis to show that. For in the small trees, usually the mortality rate is driven by the competition factors, like between the neighbor trees. But for the large trees, it's more determined by the environmental factors, especially the soil nutrient availability in this ecosystem. So in fact, I think this is a very nice paper from Nate. I think that we summarized the figure from that. It shows that currently many dynamic vegetation models are using the traits to simulate mortality rate, but they usually use so different traits to simulate mortality rate. So the algorithm is not it's changed a lot between the models. For example, we found that the LM3 PPA model used the U-shaped mortality pattern for canopy trees, as what we have seen in the data based on our site. But in many other models, like OKD is still using a constant rate, but some models using the wood density, and also like recently some models try to incorporate the carbon starvation and hydraulic failure, different mechanisms to evaluate to improve the simulation of mortality rate based on those traits. So we also did a literature survey on those empirical trait-based studies. It's about 400 studies, which studied the relationship between the plant functional traits and like carbon variables or processes. You can see that most of these studies are now work on the a published paper are focused on the carbon input, and then a few of them are focused on the decomposition processes. I think this is what I think the empirical studies need more data to study this correlation between or covariance between the plant functional traits and the decomposition processes. So I'm going to summarize my talk today. So I think the model uncertainty on land carbon cycle can be further reduced by an improved parameterization of plant functional traits. And for the first question, I think, yes, we can link the plant functional traits and the carbon process for model evaluation based on we can trace them to the baseline carbon residence time and the GPP and to some key traits. And the second is we show that the parameters of traits the uncertainty can propagate and through the carbon process, especially the loop in the vegetation. And it's still challenging, I think, to evaluate the CMIP models because we don't have much details of these parameters, especially trait parameters and the equation algorithms in these models. And for the second question, I think we still need to explore the probability distributions of different traits acclimation to the environmental factors. And also we need more data of community level trait covariance on not only the spatial scale, but also the temporal scale. And also we need models. We have many model intercomparison projects. I think we need more output of the data of the plant functional traits associated with the carbon processes. So that's all I have today. So thank you for your listening. Hi.